proteins, including serum, urine, and cerebrospinal fluid, CSF proteins, lipoproteins, isoenzymes, and so on. Electrophoresis is defined as the movement of charged molecules in a liquid medium when an electric field is applied. Zone electrophoresis is defined as the movement of charged molecules in a porous supporting medium where the molecules separate as distinct zones. Support medium provides a matrix that allows molecules to separate, for example, agarose gel, starch gel, polyacrylamide gel, and cellulose acetate membranes. Movement of charged particles through a medium depends on the nature of the particle, including net charge, size and shape, the character of the buffer and supporting medium, temperature, and the intensity of the electric field. Nature of the charged particle. Proteins are amphoteric and may be charged positively or negatively depending on the pH of the buffer solution. The pH at which negative and positive charges are equal on a protein is the protein's isoelectric point. Buffer solutions of pH 8.6 are generally used for serum protein electrophoresis. Using agarose gel or cellulose acetate at this alkaline pH, serum proteins take on a net negative charge and will migrate toward the anode. Albumin migrates the fastest toward the anode, and the gamma globulins remain closer to the cathode. Visualizing the separated analyte. Following electrophoresis, treat the support medium with colorimetric stains or fluorescent chemicals. Amido Black B, Ponzo S, and Kumasi Brilliant Blue stains are used for visualizing serum proteins. Silver nitrate is used for CSF proteins. Fat red 7B and oil red O are used for lipoproteins, and nitrotetrazolium blue is used for lactate dehydrogenase isoenzymes. Detection and quantification of the separated protein is accomplished using a densitometer. Commonly encountered problems in electrophoresis: holes in staining pattern, analyte present in too high a concentration, very slow migration, voltage too low, sample precipitates in support, pH too high or low, excessive heat production. Isoelectric focusing is a type of zone electrophoresis in which protein separation is based on the isoelectric point of the proteins. This method utilizes polyacrylamide or agarose gel containing a pH gradient formed by ampholytes in the medium. When exposed to an electric field, the ampholytes migrate based on their pI to their respective positions in the gradient. In turn, the serum proteins will migrate in the gel to the position where the gel's pH equals the pI of the respective protein. Capillary electrophoresis is based on electroosmotic flow. When an electric field is applied, the flow of liquid is in the direction of the cathode. Thus, EOF regulates the speed at which solutes move through the capillary. Hemoglobin, tetramer composed of 4 globin chains, 4 heme groups, and 4 iron atoms. Hemoglobin A1, 2 alpha chains and 2 beta chains. Hemoglobin A2, 2 alpha chains and 2 delta chains. Hemoglobin F, 2 alpha chains and 2 gamma chains. A number of hemoglobinopathies exist where a substitution of one amino acid on either the alpha chain or the beta chain causes the formation of an abnormal hemoglobin molecule. Hemoglobin S, substitution of valine for glutamic acid in position 6 of the beta chain. Hemoglobin C, substitution of lysine for glutamic acid in position 6 of the beta chain. Although hemoglobin differentiation is best achieved by use of electrophoresis, hemoglobin F may be differentiated from the majority of human hemoglobins because of its alkali resistance. At pH 8.6, hemoglobins have a net negative charge and migrate from the point of application toward the anode. Using cellulose acetate, hemoglobin A moves the fastest toward the anode, followed by hemoglobin F and hemoglobins S, G, and D, which migrate with the same mobility. Hemoglobins A2, C, O, and E have the same electrophoretic mobility and migrate slightly slower than hemoglobin S, G, and D. At pH 6.2 on agar gel, hemoglobins exhibit different electrophoretic mobilities in comparison with hemoglobins electrophoresed at pH 8.6 on cellulose acetate. Order of migration from the most anodal hemoglobin to the most cathodal hemoglobin is hemoglobins C and S, followed by hemoglobins A1, A2, D, and G, which migrate as a group with the same mobility, followed by hemoglobin F. The different migration patterns seen with cellulose acetate at pH 8.6 and agar gel at pH 6.2 are useful in differentiating hemoglobins that migrate with the same electrophoretic mobility. Centrifugal analysis. Centrifugal force moves samples and reagents into cuva areas for simultaneous analysis. Discrete analysis. Each sample reaction is compartmentalized. This may relate to an analyzer designed to assay only one analyte, for example, glucose, or an analyzer capable of performing multiple tests, where the sample and reagents are in a separate cuvee or reaction vessel for each test. Batch analysis. Samples processed as a group. Random access, able to perform individual tests or panels, and allows for stat samples to be added to the run ahead of other specimens. Standalone, instrument from a single discipline with automated capability. Automated standalone, instrument from a single discipline with additional internal automated capability, for example, auto-repeat and auto-dilute. Modular work cell, at least 2 instruments from a single discipline with 1 controller. Multiple platform, instrument able to perform tests from at least 2 disciplines. Integrated modular system, at least 2 analytical modules supported by 1 sample and reagent processing and delivery system. Pneumatic tube system, transports specimens quickly from one location to another. Throughput, maximum number of tests generated per hour.